Now let me just again define these very generic terms. These are all genetic terms, okay? And they are kind of related to the broad ideas I've been giving you about molecular biology in the first few lectures and genetics now. So you already hopefully clearly understand the relationship about information on genes and the fact that functional entities, here we are talking about proteins. Remember, RNA is also functional, okay? Uh, and you basically, this gene influences in the Mendelian world, one gene influences one trait, okay? In the modern genetics term, one gene can influence multiple traits. One gene can control uh, the length of your bones, and also the same gene can control how intelligent you are, or at least contribute to how intelligent you are. So one gene can influence many, many features, and these are not necessarily morphological features. They can be behavioral features also. At the same time, multiple genes can influence one trait. And skin color, your height are examples of where multiple genes regulate the same trait. Okay. So what I'm going to do in this slide is quickly define certain terms used by a person called Hermann Mueller many years ago, and I'll show you Mueller's face as the last slide of this talk. Okay. So a gene makes a functional polypeptide which works to give you a phenotype. Okay. Now, if the gene cannot make that functional polypeptide because of a mutation which interferes with its folding or interferes with the complete production of the folded functional polypeptide, you get a very severe mutant phenotype. And the severest mutant phenotype, if so, now I'm going to use arbitrary mathematics as I call it. Let us say, now this is a little different from what I did yesterday, but similar. Let us say that your phenotype is defined by 100 units, 100 units of whatever we are talking about. And let us assume that these 100 units are given by two alleles, and 50 plus 50 gives you 100. Fine. Now this is your wild type phenotype. Not everything is mathematical, but it is the simplest way to explain it to you. In the case of a very strong phenotype, which we call as a null, we define this as 0 units. Basically, there is no activity of the functional entity of the gene. So both alleles are not contributing. That's a null. Okay. The word amorph is also used instead of a, this looks like nuts. It's not nuts, it's null. Uh, we also use the term amorphic. That is, does not give morphology. Sometimes the allele, maybe one allele functions, the second isn't, or both alleles are giving partial functions. It gives you a milder phenotype. This we called as a hypomorph. These are all genetic terms. But basically what you have to understand is the function is not 100 units, but it is not 0 units either. So it is, let us say, in the range, roughly speaking, 10 units to 70 units. So it's functioning, but not efficient. And the last thing, which is sort of interesting, is that if you have two alleles of a gene, one allele gives you a protein, okay? The second allele has a mutation, it also gives you a protein, but this mutant protein is misbehaving, and it misbehaves not only here, it also influences the other normal protein and causes it to misbehave also. This is called as a dominant negative allele, in the sense that one allele not only doesn't contribute, it chats to the other protein which is made by the other allele, which is normal, but somehow influences it not to function. Okay, this is called a dominant negative allele. Fine. So now here is a sort of a representation. A homozygous mutant TT, okay, can be 0 because it's a null, or it can be acting as a wild type, which is either TT or it is TT. Both of these will act as wild types, because here even 50 dose is enough to give you the wild type function. Okay. If TT is both null, you will have 0 plus 0, which is 0, and you will have absolutely no function at all. So you have an amorph, which has no function. You have a hypomorph, which has partial function, and you have an antimorph or a dominant negative, in which one allele influences the other allele to not behave in a wild type and makes the phenotype worse. Is this clear? So antimorph, hypermorph, and amorph are phenotypic terms. This is what we see. And what we are trying to understand from using the term 50, 100 amount of activity is actually genotype. Fine?
Excuse me, sir.
Yeah.
So in the last one, is it are there three alleles? Like there are two green parts shown. So the wild type has, like, why is it three?
Okay. So there are cases in which a gene during DNA replication, there is a mistake and the gene duplicates. If the gene
Okay.
duplicates, you have two copies of the same gene, usually right next to each other in the genome. This is not as often as you would think. And in that kind of scenario, you basically have double one gene giving you double the amount of protein. Okay. That is called as a hypermorph. Basically,
Okay,
you're
sir.
getting more activity. So hypermorph, antimorph, hypomorph,
So can you explain hypomorph once more?
hypo.
Yes, sir.
Okay, let me try it in a different way. So if this is an XY plot, this is activity. Okay. Now one is a wild type. So one is a wild type. This is wild type, and this is what the normal activity is. If you see activity either here or here or here or here, that is two. That is a hypomorph. That is activity less than 100. Okay. Three is a here activity zero. That is a amorph or a null, zero activity. And a hypermorph goes above 100. That is number 4. So if you have more activity than normal, that will be a hypermorph.
Sir, in the hypomorph, actually nothing is going to work now, sir, because this one is
No,
giving
it's a
us the
hypomorph
top
because
of
it
the
is giving you
allele.
partial activity.
Yes, sir, but hypermorph will basically stop the functional one also to become dysfunctional, right?
No, hypermorph is more than 100 activity. Antimorph will give you less than 100 activity because one allele is influencing the other allele.
Okay.
Sir, in evolution, when we say a gene is deleted, then it's just amorph, right?
For that gene, if that entire locus is deleted, both alleles are deleted, the organism no longer has that gene. So even using the term amorph has no meaning because the gene doesn't exist, right? You can say between chimpanzees and humans, gene number 4646 is no longer there, fine? But you don't use the
Yes, sir.
term amorph because we don't have it. But if you go out in the sun and you have a mutation in both the alleles of your gene 4646, so that that gene is exists but doesn't produce functional protein, you will call it an amorph.
In case of capital T and small t, can we consider small t as amorph?
So this is a Mendelian term, right? If you have gametes like this, capital T and small t, the small t we say is a null, then it is an amorphic allele, but it's not an amorphic gene because in the next, in the plant, it will be T and T, right? This plant we can't call a hypomorph, even though 50% of the product is being formed, because the plant shows exactly the same height as a TT. So from a phenotypic view, even though one allele is not functional, you don't see a phenotype. The plant is tall. So we call it a normal wild type plant. We don't call it a hypomorph. If in non-Mendelian genetics, a hybrid between a capital T and a small t gave you a plant which was between a tall and a dwarf, somewhere over here, we could have called it a hypomorph.
So, sir, in case of incomplete dominance, this happens.
In case of incomplete dominance, you are very right. It is a hypomorphic feature. Okay.
Sir, activity of antimorph will also be 0 unit.
It will be 0 unit. It can be 0 units because one allele is trying to work. It's like twins. One twin wants to work, the other twin says, I don't want you to work. So he'll try and other twin from working. Okay. And
Okay, sir.
this means that the activity will be lower than it should be based on a single allele. And that's an antimorph.
So could you please distinguish between antimorph and hypermorph?
So in a hypermorph, so this is what we call as so-called normal activity. A wild type is this, a hypermorph is this, fine? And an antimorph is when you have protein being produced by one allele, let's call this allele X, and your other allele, X prime, is blocking the activity of this protein. So the actual activity will be low. That is a antimorph or a dominant negative. Is that clear?
Okay, sir.
So what about when both the alleles are mutated in antimorph at the first case?
No, antimorph can only work when you have one allele which is functional and one allele which is stopping the other. Both are
Sir,
working, then it's a amorph.
okay. So in the case of small t, so you said that when it's capital T, small t, we say it's amorph because it's tall, there's no effect of the small t. Small t is not producing anything.
That is one explanation, simplest explanation. Small t is dysfunctional.
But what if when small t is producing some amount, but it's not the same as capital T, but somewhere in between? Like if capital T is for red color, then small t would be for pink color or something.
Okay, you are bringing complicated things into the discussion. Let us say for a plant to be tall, there is a certain threshold of, let's say T gene codes for a growth hormone. Okay, let's say the small t we're giving you 10% activity. Fine. Together it is 20%. Now, if this 20% is below a threshold, let's call the threshold as 49%. And if you don't cross the threshold, you're not going to get a tall plant. So each allele is a hypomorph, but it is not contributing to a tall plant because the threshold has not been reached. So now we are becoming a little complicated in our discussion.
Okay, so understood.
Okay.